Our next speaker is from a little bit north of here. Yeah. Dr. Glen Stanosz is from the University of Wisconsin, and he will talk to us about tree health challenges in production of short rotation woody crops, particularly the hybrid poplar.
So thank you for the opportunity to go with the microphone. Students say I don't need to have a microphone. Thanks for the opportunity to speak with you, and I really want to thank Greenwood Resources for this opportunity. I'm here at their behest. I am a pathologist. I'll be looking at the potential for disease on some of these clones here. I've been other places, and as Randy alluded, diseases have been one of the potential limiting factors in short rotation woody crop production. And it doesn't matter if we're talking about genus populus or genus salix, the willows that have been cultivated in Europe for some time. Disease always becomes one of the problems. And I want to give you a handout, for those of you who like to read, you can read. Those of you who like to look at pictures, I put pictures. But I want to give you a little bit of philosophy. Again, I think as Randy said, when you get older, you start to philosophize a little bit more about why you see some of the things you see. And when I look at epidemics of tree diseases, we all know our native forests, our natural forests, there are diseases in the forest, but yet the world is green, right? I mean, the pine trees grow up in Wisconsin, the sugar maple grows, and diseases are there, but they're kind of in balance with that natural crop. So why do we get epidemic disease problems? And particularly, why does this apply to short rotation and intensive culture trees? Well, the first thing is scale up. And that's been alluded to. Right now we have relatively small acreages of test plantings, but it is an absolute rule that when you scale up a crop, and I was trained in a crop plant pathology department, even though I worked on trees, but when you scale up any crop, you scale up what we call the target tissue. When you vastly increase the amount of target tissue, the diseases that can take advantage of that tissue take advantage of it. I was thinking of a good analogy for this. Randy likes to hunt deer, and I suppose if we scaled up the number of deer that trotted in front of his gun barrel, he might get one, But I mean, that is a general rule in all of plant pathology. When you scale something up, you're gonna scale up the diseases that exist. And that's especially true if you're dealing with something with limited genetic diversity. A really good example of that from the south here was scale up of loblolly pine. After World War II, loblolly pine became the tree of choice, and it's been planted by the millions and millions and millions. What disease of loblolly pine became the scourge of loblolly pine? It's been taken care of pretty much now, but fusiform rust. Fusiform rust was relatively unknown before loblolly pine was scaled up. Native rust, native tree, and it became a huge problem. The second thing that all of us should be acutely aware of is introduction of new pathogens from other countries. And this has happened in North America just again and again and again. As a northern forest pathologist, we're really aware of Dutch elm disease, white pine blister rust. Some of you maybe have heard about a butternut disease that has recently apparently moved from western United States into the east. What's the big one down here? On red bay, you've got a red bay disease now in the southeast. Laurel wilt, exotic fungus that probably came into the port of Savannah and it's killing red bay. Very aggressive pathogens that kill native hosts because they've come from some other environment, and our native hosts have never seen those pathogens before. They have developed no resistance to those over time. Well, the corollary to introducing a native or introducing an exotic pathogen to a native host, the corollary is when you introduce an exotic host to a native pathogen. And that's been one of our issues with hybrid poplars, because we're taking exotic trees from Asia or from Europe, and we're bringing those to North America, and they're meeting pathogens that they've never seen before. So it's all part of that same kind of thing of new host pathogen combinations. And then the last thing that we see as a common factor in these epidemics is when we put trees in a situation or utilize practices that really tip the balance toward the pathogen and really enhance the potential for disease development. And there it is. Plant single genotypes close together, and yeah, you give the pathogen quite a distinct advantage. And I've just been getting leaf samples from colleagues up at University of Minnesota Duluth, and they sent me samples from 2 different areas where their program is growing hybrid poplar. In one set of samples there's hardly any disease, the other set of samples was just loaded with the disease. And I said, what's the difference? Well, the one location that was just loaded with the disease, every sample was covered, very tightly spaced, closely spaced trees, very little ventilation. Trees get wet, they stay wet for a long period of time. They have a situation, they've enhanced disease development. So, what do we do about that? Well, if we're in modern agriculture, how do you think we control potato diseases up in Wisconsin where we grow? Forget them Idaho potatoes, it's Wisconsin potatoes that you should be looking for. Well, the crop dusters spray those potatoes every 7 to 10 days. It's one of the most unsustainable production systems that we have. But we all like our fries and our chips and our baked potatoes with good Wisconsin sour cream, That's what we like. So they come, they spray those fields. Well, that's just not feasible for us even in this kind of intensive production system. We're not going to spray fungicides on these things every 7 to 10 days to try to control diseases. We might have some limited use of insecticides if there's not a better answer for that. But in forestry, we just don't do aerial spraying. We can in a nursery. In a nursery production situation, we can very efficiently control the diseases with fungicidal sprays. So we have tools like that, but it's an economic reality of big plantations. So what do we have to do? And Randy's alluded to this, and people like Randy and the other people who are breeders, they're the guys with the answers. And we know in woody crop production that selection of superior genotypes is the way that we can control the disease problems. And again, I can come back to loblolly pine. After fusiform rust became a big problem in loblolly pine, breeders sat down with pathologists, they got their heads together, they developed selection programs. Most of the commercially sold loblolly pine comes out of families that have been screened for their resistance to the fusiform rust, and even screened for resistance to the local populations of fusiform rust, because there are different populations of fusiform rust in East Texas than there are in Georgia, than there are in North Carolina. So smarter people than me, like Randy Russo, that do the genetics and the breeding, these are the people that have the answers for us. So as pessimistic as I might be, and usually people look at forest pathologists and say, boy, you guys are a bunch of, you're depressed all the time, you're looking at dead stuff, and you get excited when you see the trees die. And no, I'm really on the tree side, but the take-home message that I want to give you is that it's the genetics and the selection of the superior material, not just for growth rate, but for resistance to disease, that is the important part of this process. And that's why you see these genotype trials down here. And Greenwood is kind of one of the points of the spears of trying to figure out, do we have genetic resistance in hybrids where we can bring in growth characteristics, but we can also bring in disease resistance. And then I guess two final points. One thing we talk about is durable resistance. The ag crop people talk about durable resistance, meaning they want a soybean or a corn that they can plant year after year, that same variety, or a variety with at least the same resistance genes, and that resistance is going to be durable. Well, it's even more important for us, because we're going to put a genotype out, and either it's going to be a timber tree that's going to grow for maybe 12, 15, 16 years, and that resistance has to be maintained through that entire rotation, or a coppice that you're repeatedly cutting and resprouting the same genotype over large acreages, it's a real challenge to have that durable resistance. If resistance breaks down halfway through a poplar rotation, you just can't switch to a different variety like the soybean farmer or the corn farmer. So that is going to be a challenge, and it's going to require screening not just for short periods of time, but watching through a whole production cycle. And then the final point that I'd like to make, and probably Randy and I will argue over the beer about this, but when a plant pathologist talks about screening, they're not talking about merely making a selection. So when you plant a bunch of trees out, and I watched people in Wisconsin do this, planting hybrid poplars for years, they'd plant them out in trials, and then they'd wait to see which ones die. And then they'd say, okay, the ones that didn't die, that's our selection. Well, I don't call that screening. I think screening is when you put the host and the pathogen together, and it's a well-characterized pathogen. You know what it is genetically just as much as you know what that tree is genetically, and you put those together in a conducive environment so that disease will occur. Okay, I once, as a graduate student, they showed us a wheat screening operation, and they had a line of susceptible wheat that they knew was going to get diseased, and then a line of wheat they were testing, and then a line of wheat that was susceptible, and a line of wheat they were testing, and they brought in the inoculum, and they inoculated those susceptible wheat rows that they called spreader rows, and they had overhead irrigation to make sure that the leaves would stay wet and get infected, and they created that epidemic, and then they could make their selection. So they're screening under a heavy disease pressure. In addition to that kind of screening under heavy disease pressure, boy, I've argued with other people, not Randy, other people, that when you do that, you need to have a yardstick of some kind. So I've had a grad student very, very successfully screen for Septoria canker resistant, and we included a resistant standard and a susceptible standard. So we had our yardstick. And every clone we tested, we could see where it would fall between that resistant standard and that susceptible standard. We could see where in the continuum it was. So that's something that we really need to think about and incorporate into trials over the longer term. I'm absolutely adamant about that you got to know how good your stuff is, or it might merely be what plant pathologists call an escape. Maybe you just didn't have disease pressure there, so you don't know how good it is. But anyway, on the back I showed you some of my favorite organisms. Melamsura rust, you see a little Melamsura rust. Native species is Melamsura medusae. Unfortunately, we know we've had one exotic Melamsura species introduced to the United States, and damn thing won't stay to itself. It's actually started to hybridize with our native rust, so that's kind of a crapshoot now. We have to characterize what rust we're dealing with in different places. Marsanina leaf spot, something I'm very interested in, the middle two pictures. Marsanina, in North America, we think we have 3 species and 2 different subspecies of one of those species, but we've never done a modern molecular test to be able to know what we have. I'm starting to make some collections, and hopefully we'll know which Marsanina we have in the Pacific Northwest, in the Southeast, in the Lake States, or again, are we sharing those as we move material around? Marsanina, interestingly, we've got some people doing research in Wisconsin who have completely lost research plots. The Marsanina is so bad it defoliates the trees, and they've been killed. And we never used to see that 15 years ago, and we're concerned that maybe we've gotten some new Marsanina. Probably one of Randy's damn things came up. I don't know. But, that's always a concern, is that, we have invasive plants. We also have fungi that move around. Upper right there is a Venturia shoot blight. Interesting disease, does affect our native aspens up in the Northeast. Usually trees grow out of Venturia blight, but again, under conducive conditions, it could be real bad. Doesn't express itself every year. Some years we see it, some we don't, depending on rainfall. And then down at the bottom should be the scary pictures. Septoria leaf spot, it really has been the limiting factor in the eastern United States in trying to develop a hybrid-based industry. Randy said our native cottonwood is very resistant. Up in the lake states, our quaking aspen, big-tooth aspen are very resistant. But when we hybridize to bring in other desirable characteristics, which in some cases we need rooting, we need the height growth that we can get from some of these exotic poplars. Unfortunately, we can bring in disease susceptibility. But again, I want to emphasize with inputs, we can get past these. I was once told when I had a grad student start a screening project by another person who'd done poplar research that we'd never be successful, we'd never find resistance, and it wouldn't be durable anyway. And I just point to a clone. Everybody in the poplar business has heard of a clone called DM34 or Eugenii. It's not the fastest growing tree in the world. Up in Wisconsin, we call it the banker's clone. You know, it's kind of a conservative clone. Grows pretty good. We sacrifice some growth rate compared to other things. But Eugenii, which is a DN, right, Randy? DN? It's a deltoides, our native deltoides by nigra. That tree has been grown in many different states for decades and decades, and it still doesn't get septoria canker. So I think that's just an accidental example that we can solve these problems. We can have our cake and eat it too. Whoops. And I guess it's time to stop. But I'll take questions if anybody has any questions. And again, don't be scared of your pathologist. It takes a lot of courage for people who are growing trees to invite me to come and look at your trees. But again, I insist these are just little challenges. Does anybody think that we're planting anything when we plant corn that looks like unimproved ancestral corn from Mexico? No. We've solved those problems by breeding and selection and overcoming our disease problems. So, any questions? Be happy to chat quietly. Yeah?
Question on septoria.
Yeah.
I have read somewhere that if you have a septoria leaf spot, there's a guarantee that you will have canker on the stem. Is that true?
No, there's not a one-to-one correlation. So his question is about if you have septoria leaf spot on the leaves, will you get canker on the stem? I don't think there's a one-to-one correlation. I don't think susceptibility to bad leaf spot, which might defoliate you, means necessarily you're going to get severe cankering. So I don't think rating for leaf spot is sufficient if you were trying to make selections. There is some kind of correlation or some kind of relationship. I mean, if a tree doesn't get either one, it's obviously got the genes for resistance to both, or it lacks a gene for susceptibility. And I also think that we have to get a little more sophisticated about how we rate canker too, because some cankers are nothing more than superficial blemishes, and a tree can get canker, and just saying it has canker doesn't mean it's not a useful tree. It depends what that end product is. It depends how long you're going to grow that tree. The interesting thing, though, I guess from the pathogen side, is we see no difference in the pathogen between isolates that we make from the leaf spots and isolates that we make from the cankers. So from the fungus side, one genotype of the fungus is capable of doing both. But on the tree side, there's quite a range of differences, and I think we need to come up with a canker disease rating scale. I've rated canker disease in the field, and one is for presence of cankers, numbers of cankers, but then another is for the impact of those cankers. So again, if every human genotype was thrown out just because we had a freckle, there wouldn't be many humans around. And there are poplar hybrid genotypes that will get stem infections, but they can be pretty superficial. So, yes, Tim.
Glen, in the case of the willow, you alluded to willow, and frankly, there's a lot of interest with the poplar of looking at a coppice kind of management system where, maybe a two-year cycle. I guess what I'm wondering if you see the disease presence in in one rotation, does that guarantee that it will return or intensify, or in that sort of production system?
If we're talking about Septoria canker, let's just talk about that one.
Okay.
I think Septoria canker, as I've seen it, that we've grown it in my experimental farms or other places I visited, the leaf disease only intensifies over time. Many of these pathogens overwinter on the leaf litter, and in the spring they produce a primary spore up from that leaf litter that infects the leaves. And then once you get lesions on the leaves, they start sporulating right on the leaves, and you get a secondary spore that we call a repeating spore. So in nice wet summer weather, you can get 8-10 disease cycles a summer. So you see intensification just in one season, but we've also seen intensification over time. And one of our university farms, my grad student that was doing screening work, we set up his trial. The first year we didn't see a Septoria leaf spot or canker on anything. Second year we started to see leaf spot. Then we started to see naturally occurring cankers in addition to the ones that we had created by inoculating trees. Many of the leaf diseases, I think, will intensify over time. What their ultimate impact will be in a coppice situation depends. Some clones, and again, I wouldn't say for any of these diseases, we probably don't want to select for complete absence of disease either. A little bit of disease is okay, probably doesn't interfere that much with yield. How coppice will affect the impact of these diseases, or how these diseases will impact repeated coppice, though, that I don't really know. Again, you want to feed that root system to make sure you keep getting that coppice. What in the future I'm quite concerned about is something like Septoria musiva, which does invade the woody tissue. It is not limited to the bark. We did histological work to show it actually does penetrate into the wood tissue. When you have a coppice, you're creating those wounds every year or two, and I'm really concerned about what fungi are going to think that's the dinner bell ringing. Mm-hmm. All those wounds down there at the coppice. We don't know because who's done it and done it repeatedly. So that's something that if I had, all the time and money in the world, professors are full of ideas. Mm-hmm. We just lack the wherewithal and smart grad students to come up with all the answers for us. So, anyway, I'd be happy. Oh, yeah, Rick.
So you want to relate what about endophyte work on? Because we've got a couple, Doty and Newcomb, working with endophytes, and some of it's fungal prevention. How about that? What's your thoughts on that?
I'm counting to 10. I'll just repeat what I told someone. I actually told someone at the meeting up in Seattle. Okay, just to explain to everybody what endophytes are. So there are microorganisms that will infect plants, reside in the plant, be it the root tissue, the bark tissue, sometimes even the wood, and the leaves. Leaves are full of these organisms that, in general, when we say endophyte, it produces no symptom. In other words, they're not pathogens. I draw the line. If it's a pathogen, I call it a pathogen. If it's an endophyte, meaning it can infect the plant tissue, it can live there and reside in that plant tissue, but doesn't cause any symptoms. So different endophytes have been shown to have different sorts of impacts. But what I said at the meeting in Seattle was no endophyte is going to turn a susceptible clone into a resistant clone. It's not going to happen. And I will go to my grave saying that that's true. In particular, if somebody critically looks at the research, a lot of that research is done in a greenhouse or under extremely artificial conditions. For example, if I isolate an endophyte from a leaf, and that means it was present in that leaf. Maybe there was one colony, maybe there were 10 colonies, these tiny little microscopic colonies. Now I bring that endophyte into my lab, I batch it up in culture, I spray 10 to the 5th, so what is that, 100,000 spores per mil onto the leaves, and then I inoculate with a rust fungus and I see less rust on that plant. What the hell does that have to do with that plant growing out in the field? Absolutely nothing. So when I came to UW-Madison about 20 years ago, we were spending millions of dollars on biocontrol research, in which you find your magic organism, you spray it on the plant, and you're going to, some disease isn't going to happen then because of that. Works great in a growth chamber in the basement of Russell Labs at UW-Madison. Never been adopted in the field for field crop disease control. So I'm not saying that we shouldn't study endophytes. Some of the differences that we see between clones in the field may be due to the fact that some of them are harboring endophytes in their leaves and others are not. But I'm thinking that it's around the margins, okay? But it's not going to completely change the phenotype, meaning what we see. It's not going to completely change it. On the other hand, one of the real interesting things that people have documented, that if you go into plantings where there are mixed plantings of hybrids and native trees, the native trees typically have a higher diversity of endophytes in their leaves and a higher density of endophytes in their leaves than the hybrids. Now, why would that be? Well, if that hybrid has an exotic parent, that exotic parent hasn't co-evolved with those endophytes and hasn't picked up the ability to harbor those endophytes. But again, I think, boy, I wouldn't put my retirement money into that hypothesis that endophytes are somehow going to change susceptible material into resistant material. I'd go to the bank with that. Around the margins, yes, but again, some of the work that I've seen tells me it's so far removed from reality, and I can prove all kinds of things in growth chambers with potted trees with 10 to the 5th spores per mil, but that doesn't tell me that what you're seeing is got any relationship with the presence of that fungus or the activity of the fungus in the field. So I've been a skeptic of what's been going on out there. I've seen it. But yeah, I'm a bit skeptical. And again, it's because of my experience with biocontrol research. We have organisms at UW-Madison that were patented because, we got this magic microbe and it's going to prevent soybean diseases and this disease and that disease. None of that ever came to market. None of that's ever been used. So it's neat biology. And again, around the margins might be pretty interesting. We also know, for example, with rust, there are organisms on cottonwood leaves that parasitize the rust fungus. So the parasitic rust fungus has a parasite. And heck, that's great too. That people have shown that actually the parasitism of those little rust pustules actually reduces the rate of sporulation of those rust pustules. So if you could figure out how to foster that, you wouldn't get rid of rust, but around the margins again, you'd suppress it. So please don't say I'm telling bad stories about endophytes. They're real, they're out there, they're probably part of the reason why some of our native trees behave the way they do when certain pathogens meet them. But I think the stuff Randy does in terms of seeing superior genotypes and selecting those is going to be a much bigger payoff because they've got the inherent genes for resistance, and that's what we're looking for. Sorry I rambled on so much, but as a professor, I'm used to 55-minute blocks.